Hello, my dear students of class 11 science. Welcome to biology class. We are dealing with the topic mechanism of photosynthesis. In the last class we have learned about the light dependent reaction or Hill reactions or photochemical phase of photosynthesis. Clear? Now today we learn about the second mechanism of photosynthesis, that is called light independent reaction or dark reaction or biosynthetic phase or Blackman reactions or carbon dioxide fixation. Okay, so today we are going to learn about the light independent reaction or the dark phase of photosynthesis. So this light independent reaction or the dark phase of photosynthesis, it occurs in the stroma of chloroplast. Clear? So it utilizes the ATP and NADPs for the fixation and reduction of carbon dioxide to form carbohydrates. Now dark reaction does not mean that it usually occurs at night or in the absence of light. Clear? But usually say that it is independent of light and can occur in light or in dark. Clear? It doesn't mean that it usually occurs at night or in the absence of light. It can occur during the daytime also in the presence of light, but it's just independent of light. Okay, so it can occur in light also or in dark also, but it does not use the light energy. Clear? And in this particular phase, carbon dioxide is reduced to sugar. Okay, and whatever the enzymes that are essential for the fixation of carbon dioxide and that essential for the formation of sugar are all present in the stroma of the chloroplast. That's why the site for light independent phase of photosynthesis is the stroma of chloroplast. Now let's talk about the carbon dioxide fixation types. So generally we have got three types of carbon dioxide fixation depending upon the different plants, but we have to only go through the Calvin cycle in C3 plants and the Hertzsprung cycle in the C4 plants. Though we have got three types, okay, which are Calvin cycle in C3 plants, Hertzsprung cycle in C4 plants, and the CAM cycle in CAM plants. But we'll only go through the two process, that is Calvin cycle in C3 plants, and we will learn about the Hertzsprung cycle in C4 plants. So let us begin with the first one, that is Calvin cycle or the Calvin-Benson cycle or C3 cycle. So the Calvin cycle or the C3 cycle, it was mainly discovered by Calvin and his co-worker. Okay, so this thing developed by Calvin and his co-worker. Clear? And since we're talking about the carbon dioxide fixation, the primary acceptor molecule, very carefully, okay, the primary acceptor molecule of the cycle was found by Basham in 1955. That means the primary acceptor molecule that will fix the carbon dioxide in C3 cycle was mainly found by Basham, and that particular acceptor molecule was ribulose 1,5-biphosphate, also called RUBP. Now since these primary acceptor molecules for the carbon dioxide fixation was discovered by Basham, it's also known as Calvin-Benson cycle. Okay, so these reactions are also known as path of carbon dioxide fixation, C3 cycle, Calvin cycle, or Calvin-Benson-Basham cycle. And for this particular cycle, Calvin was awarded the Nobel Prize also in the year 1961. Okay, so let us talk about the reactions of the Calvin cycles. So Calvin cycles occurs in all the photosynthetic plants, okay, whether they have got the C3 pathway or they have got the C4 pathway. Clear? But generally over here we are dealing with the C3 cycle. Clear? But it occurs in all the photosynthetic plants that goes the C4 cycle or to the C3 pathway. Okay, now in Calvin cycle, the first product of carbon dioxide fixation is a 3-carbon acid called phosphoglyceric acid. Kindly remember this thing. Okay, the first product of carbon dioxide fixation in Calvin cycle or in C3 cycle is a 3-carbon acid called phosphoglyceric acid. So this is very important. Clear? So the first product of carbon dioxide fixation in Calvin cycle or C3 cycle is a 3-carbon acid called phosphoglyceric acid. So now we'll go in detail and we'll look at the different phases of Calvin cycle. So Calvin cycle is completed in 3 phases. The first one is called carboxylation, the second one is called reduction, and the third one is called regeneration of RuBP. Remember, RuBP is the primary acceptor of carbon dioxide in C3 cycle. So there are three phases of Calvin cycle. First one is called carboxylation, second one is called reduction, and the third one is called regeneration. So let's go to the first one, which is called the carboxylation phase. Okay, so what are the events that occur during the carboxylation phase of Calvin cycle or the 3-3 cycle? Okay, so basically what do you mean by carboxylation? So addition of carbon dioxide to a compound is called carboxylation. Clear? So addition of carbon dioxide to a particular compound is called carboxylation. Now since we are dealing with the Calvin cycle, now let's see what are the events that occurs during carboxylation in C3 cycle or in Calvin cycle. Now in Calvin cycle, a 5-carbon sugar called ribulose 1,5-biphosphate, also called RuBP. Listen very carefully. Okay, so in Calvin cycle, a 5-carbon sugar called ribulose 1,5-biphosphate, also known as RuBP, it undergoes carboxylation by the addition of carbon dioxide. Clear? So this RuBP undergoes carboxylation by the addition of carbon dioxide, and it forms 2 molecules of 3-PGA. PGA means phosphoglyceric acid. If you remember over here that I told you that the first product of carbon dioxide fixation is a 3-carbon acid called phosphoglyceric acids. So during carboxylation, this RuBP undergoes carboxylation by the addition of carbon dioxide, and it forms the 2 molecules of 3-PGA, that is phosphoglyceric acid. Okay, and the reaction is catalyzed by the enzyme called RuBP carboxylase. So remember the name of the enzyme, which is called RuBP carboxylase. So RuBP here is the compound which undergoes carboxylation by the addition of carbon dioxide to form 2 molecules of 3 phosphoglyceric acids, and this particular reaction is triggered by the enzyme called RuBP carboxylase. Okay, now since this RuBP carboxylase enzyme also has got oxygenating activity, it is also called ribulose 1,5-biphosphate carboxylase oxygenase enzyme, or in short we will call it Rubisco. Okay, since it has got oxygenating activity, so this RuBP carboxylase is also called Rubisco. That means ribulose 1,5-biphosphate carboxylase oxygenase. Clear? So let's see the reaction. What is that? The RuBP, okay, that means the 5-carbon sugar compound, it undergoes carboxylation by the addition of carbon dioxide in the presence of the enzyme Rubisco to form an intermediate compound called 2-carboxy-3-keto-1,5-biphosphoorbital. This is the intermediate compound. Clear? Now this intermediate compound will split in the presence of water molecule. Listen carefully. The intermediate compound is 2-carboxy-3-keto-1,5-biphosphoorbital. Now this particular intermediate compound will split in the presence of water to form 2 molecules of phosphoglyceric acid, which is the first product of carbon dioxide fixation in C3 cycle. Clear? Okay. Now let's go to the second step, which is called the reduction. Now in reduction there will be formation of glucose. Okay, so in reduction phase there will be formation of glucose molecule. So let's talk in detail. So this reaction, okay, that means in this reduction phase of C3 cycle or the Calvin cycle, the PGA, that is phosphoglyceric acid, which is formed during the carboxylation phase, will be phosphorylated by the ATP. Okay, so this PGA will be phosphorylated by the ATP with the help of the enzyme triose phosphate kinase, and it will give rise to 1,3-biphosphoglyceric acid. So look over here. So in the presence of the enzyme phosphoglycerate kinase, clear? Listen very carefully. In the presence of the enzyme phosphoglycerate kinase, the phosphoglyceric acids will be phosphorylated by the ATP to form 1,3-biphosphoglyceric acid. Clear? Okay. Now in second one, this 1,3-biphosphoglyceric acid, Listen carefully. This 1,3-biphosphoglyceric acid will be reduced by NADPH. Can you see over here that 1,3-biphosphoglyceric acid will be reduced by NADPH in the presence of the enzyme triose phosphate dehydrogenase into glyceraldehyde 3-phosphate. Clear? Now, this glyceraldehyde 3-phosphate, it undergoes isomerization in the presence of the enzyme triose phosphate isomerase to form dihydroxyacetone 3-phosphate. Okay. Now, in the fourth one, the two isomers of glyceraldehyde 3-phosphate and dihydroxyacetone 3-phosphate, it will condense in the presence of the enzyme aldolase to form fructose 1,6-biphosphate. Again, one more time, the two isomers, that is glyceraldehyde 3-phosphate and dihydroxyacetone 3-phosphate, in the presence of the enzyme aldolase will condense to form fructose 1,6-biphosphate. Now, this fructose 1,6-biphosphate, listen carefully, loses one phosphate group. Okay, and in the presence of the enzyme phosphatase, it will form fructose 6-phosphate. So this fructose 1,6-biphosphate will lose one phosphate group in the presence of the enzyme phosphatase and forms fructose 6-phosphate. Now finally, this fructose 6-phosphate will change into its isomer called glucose 6-phosphate. Okay, and glucose 6-phosphate will form the glucose. So you can see over here that fructose 6-phosphate will will form its isomer in the presence of the enzyme isomerase to form glucose 6-phosphate, and glucose 6-phosphate will form the glucose by the help of the enzyme phosphatase. So glucose is the product of reduction phase of Calvin cycle or the C3 cycle. So what are the events that occur during the reduction phase of Calvin cycle or the C3 cycle? Listen very carefully. The phosphoglyceric acid which was produced in the carboxylation phase of C3 cycle will be phosphorylated by the presence of ATP and by the help of the enzyme triose phosphate kinase to form 1,3-biphosphoglyceric acid. Now that 1,3-biphosphoglyceric acids will be reduced by NADPH in the presence of the enzyme triose phosphate dehydrogenase to form glyceraldehyde 3-phosphate. Okay, and glyceraldehyde 3-phosphate undergoes isomerization to form dihydroxyacetone 3-phosphate in the presence of the enzyme triose phosphate isomerase. Now the two isomer, that is glyceraldehyde 3-phosphate and dihydroxyacetone 3-phosphate, will undergo condensation in the presence of the enzyme aldolase to form fructose 1,6-biphosphate. Now this fructose 1,6-biphosphate will lose one phosphate group and it forms the fructose 6-phosphate, and finally the fructose 6-phosphate will form its isomer, that is glucose 6-phosphate, and glucose 6-phosphate will finally form the glucose. So glucose is the product of reduction phase of C3 cycle, or we can say the Calvin cycle. Now let's talk about the last phase, which is called the regeneration phase, where there will be regeneration of RUBP, the enzyme, which is the carbon dioxide acceptor molecule. Okay, so here what I mean is that the fructose 6-phosphate and the glyceraldehyde 3-phosphate. Okay, look over here, the first one. So in this particular regeneration phase of C3 cycle, the fructose 6-phosphate and the glyceraldehyde 3-phosphate, they will react to form erythrose 4-phosphate and xylose 5-phosphate. So these are the products which will be formed from the fructose 6-phosphate and the glyceraldehyde 3-phosphate by the help of the enzyme transketolase, where TPP is the cofactor. Okay, now in next one, the erythrose 4-phosphate and dihydroxyacetone 3-phosphate will condense to produce pseudoheptulose 1,7-biphosphate in the presence of the enzyme called transaldolase. Now in the third step, the pseudoheptulose 1,7-biphosphate, okay, this particular product will lose one phosphate group and it will form pseudoheptulose 7-phosphate in the presence of the enzyme phosphatase. Clear? Now the pseudoheptulose, in the fourth one, the pseudoheptulose 7-phosphate will react with 3-phosphoglyceraldehyde or glyceraldehyde 3-phosphate and it will form ribose 5-phosphate and xylulose 5-phosphate. Okay, and this reaction is triggered by the enzyme transcatolase in the presence of the cofactor TPP. Now, the ribose 5-phosphate is converted into ribulose 5-phosphate in the presence of the enzyme phospho-ribose isomerase. So that means finally the ribose 5-phosphate is forming the ribulose 5-phosphate. Ribose 5-phosphate in the presence of the enzyme phospho-ribose isomerase is forming the product ribulose 5-phosphate. Now the xylulose 5-phosphate will be isomerized to form ribulose 5-phosphate in the presence of the enzyme epimerase. Okay, so finally ribulose 5-phosphate is phosphorylated by ATP to produce ribulose 1,5-biphosphate, or we call it Rubisco, and this is mainly triggered by the enzyme called phosphorubicose. So finally the carbon dioxide acceptor of the C3 cycle or the Calvin cycle, that is ribulose 1,5-biphosphate or Rubisco, is regenerated. Okay, so this is the third phase of Calvin cycle of the C3 cycle, where there will be regeneration of ribulose 1,5-biphosphate or Rubisco, which is the carbon dioxide acceptor molecule or the enzyme. Okay, so the overall reaction you can see over here, we have got 6 molecules of carbon dioxide plus 6 molecules of RUBP, 18 molecules of ATP and 12 molecules of NADPH. In the presence of different enzymes, there will be formation of glucose C6H12O6, there will be regeneration of RUBP, there will be production of ADP, because whenever ATP will be utilized, there will be formation of ADP. Okay, so these are the products or the oral reactions of C3 cycle of photosynthesis. Now, how many molecules of ATP and how many molecules of NADPH are utilized for the formation of one molecule of glucose in C3 cycle? That is the most important question. Okay, so one more time, how many molecules of ATP and how many molecules of NADPH are required for the formation of one molecule of glucose in C3 cycle or in Calvin cycle? That is the most important question. Okay, so hence for every carbon dioxide molecule that enters the Calvin cycle, 3 molecules of ATP and 2 molecules of NADPH are required. Okay, so for each and every carbon dioxide molecules that enters the C3 cycle or the Calvin cycle, 3 molecules of ATP and 2 molecules of NADPH are required. Okay, to make one molecule of glucose, 6 turns of Calvin cycles are required. Look over here. To make one molecule of glucose, how many turns of Calvin cycle required? 6 turns of Calvin cycle are required. So therefore, for each molecule of glucose to be produced, how many molecules of ATP will be required? 6 into 3 will be 18, because for every carbon dioxide molecule that enter the Calvin cycle or C3 cycle, 3 molecules of ATP will be required. So for the formation of one molecule of glucose, 6 turns of Calvin cycle has to be done. Okay, so 6 into 3 will be how much? 18. So 18 molecules of ATP will be required. And how many molecules of NADPH will be required for formation of one molecule of glucose? 6 into 2 will be 12. So therefore, for each molecule of glucose, 18 molecules of ATP and 12 molecules of NADPH are required in C3 cycle or Calvin cycle, because 6 turns of Calvin cycle are required for the formation of one molecule of glucose. Okay, so this is the overall reactions or the process of C3 cycle or the Calvin cycle. Okay, now let's talk about the next one, that is the C4 pathway or the C4 cycle, or we can say the Hatch-Slack cycle. It's in the page number 13.21. Okay, so C4 pathway was discovered by M.D. Hatch and C. Hatch-Slack in 1966. So over here, what is the product that is being produced? Okay, so mainly in this pathway, a 4-carbon compound called oxaloacetic acid is formed, OAA. A 4-carbon compound called OAA, oxalic acetic acid, is formed, and hence it is called the C4 pathway. If you remember, in the C3 pathway we have got the phosphoglyceric acid, 3-carbon acid, which is called the phosphoglyceric acid, and hence the pathway is known as C3 pathway or the C3 cycle. And in C4 pathway, the product will be a 4-carbon compound called oxaloacetic acid, which is mainly formed by the initial fixation of carbon dioxide. So hence the pathway is known as C4 pathway, and also called as the Hatch-Slack cycle after the name of its discoverers. Okay, so let's see occurrence. It occurs in tropical and in subtropical grasses like crabgrass, okay, maize, sugarcane, fuel, millet, and sorghum, all monocot and some dicot also. Now let's go into the reactions or the featured C4 plants. These plants are mostly found in the tropical and the subtropical regions with high temperature, aridity and salinity. They show response to high light intensities. They have greater productivity of biomass. They have a special type of leaf anatomy called the crunch anatomy, which we'll learn afterwards what does crunch anatomy means. Okay, then they can tolerate high temperature and they lack photorespirations. Okay, so what does crunch anatomy means? Because we are saying that the C4 pathway occurs in a particular type of leaf having a special anatomy called the crunch anatomy. So what does crunch anatomy means? Okay, so the anatomy of leaves of C4 plant is different from that of the C3 plant. So the anatomy of C4 plant and the C3 plant, or the leaf of the C4 plant and the leaf of the C3 plant, is different, because in C4 plant or in the leaf of the C4 plant we are getting the crunch anatomy, and in such leaf the palisade tissue is absent. If you remember, we have learned in the anatomy of the leaf that the mesophyll cells or tissue divided into palisade and the spongy mesophyll cells. Okay, so here in crunch anatomy the palisade tissue is absent. Okay, so in the absence of palisade tissue, the mesophyll cells forms a ring outside the bundle sheath. Can you see over here this ring? So this particular ring is formed by the mesophyll cells. So mesophyll cells forms a ring outside the bundle sheath around the vascular bundle. So outside we will find the ring of mesophyll cells. Okay, and inside we will find the bundle sheath cells along with the vascular bundles, that means the element phloem. So in such plants palisade tissue is absent and mesophyll cells forms a ring outside the bundle sheath around the vascular bundle, and this type of anatomy is called crunch anatomy. Okay, so here we have seen that this particular ring is mainly formed by the mesophyll cells, which is formed outside the bundle sheath cells and around the vascular bundles. Okay, so now let's see the biochemical pathway of C4 cycle. So this pathway is also a cyclic process, okay, where there will be double fixation of carbon dioxide. We have got initial fixation and we have got the final fixation of carbon dioxide in C4 pathway. So what are the initial product of carbon dioxide fixation and what are the final product of carbon dioxide fixation in C4 pathway? We are going to discuss about that. Okay, so initial fixation of carbon dioxide occurs in mesophyll cell, that means in a ring. Okay, so initial fixation of carbon dioxide occurs in a mesophyll cells, the ring outside the bundle sheath. Okay, so the carbon dioxide is first fixed in the ring of mesophyll cells around the bundle sheath, and this fixation is mainly done by a 3-carbon compound called phosphoenol pyruvic acid, PAP. Okay, so this PAP or PP, we also call the PP carboxylase. So this is the name of the enzyme, PP carboxylase. So here in case of initial fixation of carbon dioxide in C4 pathway, it occurs in the mesophyll cells. Okay, and the carbon dioxide is mainly fixed by a 3-carbon compound called phosphoenol pyruvic acid, and that will lead to the formation of a compound called oxaloacetic acid, also known as OAA, and therefore the name of the enzyme is PAP carboxylase. Done. Listen very carefully that the initial, that in biochemical pathway of C4 cycle, that carbon dioxide fixation occurs in 2 phases, the initial phase and the final phase. So in initial fixation of carbon dioxide, it occurs in the mesophyll cells, okay, that forms a ring outside the bundle sheath around the vascular bundle. Okay, so therefore the carbon dioxide fixation in initial part is mainly the phosphoenol pyruvic acid that will lead to the formation of a 4-carbon compound called oxaloacetic acid. Hence the pathway is called C4 pathway, and this particular reaction is triggered by an enzyme called PAP carboxylase. Now the oxaloacetic acid that has which is the initial product of carbon dioxide fixation in the mesophyll cells, it will be reduced to malic acid or it will be transaminated to aspartic acid. Clear? So oxaloacetic acid, it will be reduced to form the malic acid by the help of the enzyme malate dehydrogenase, or it will be transaminated by the enzyme called transaminase to form the aspartic acid. Clear? So number 2 is transferred. So malic acid or aspartic acid is translocated to bundle sheath cell through the plasmodesmata, and inside the bundle sheath cells malic acid is decarboxylated and aspartic acid is deaminated. So look over here. So this is the mesophyll cells. Okay, so in this particular portion the initial fixation of carbon dioxide occur, where the product will be this 4-carbon compound called oxaloacetic acid. After the formation of that oxaloacetic acid, it will be either reduced to form the malic acid or it will form the aspartic acids. So whatever the products will be formed, now it will be transported through the help of plasmodesmata to the bundle sheath cells. Okay, so the product of the initial fixation of carbon dioxide in the C4 pathway, which occurs in the mesophyll cells, that is the initial fixation, that product now will enter into the bundle sheath cells by the help of plasmodesmata. Clear? Now, what I mean is that inside the bundle sheath cells, malic acid is decarboxylated by the help of the enzyme decarboxylase to form the pyruvic acid, or the aspartic acid will be deaminated to form, look over here, aspartic acid will be deaminated to form alanine and oxaloacetic acids. Clear? Okay. Now, third one is final fixation of carbon dioxide in the bundle sheath cell. That means over here in the bundle sheath cells, the final fixation of carbon dioxide occur, and that fixation of carbon dioxide in the bundle sheath cell takes place by the help of C3 cycle. Clear? So here you can see that the C3 cycle is occurring in the bundle sheath cells. Clear? So the carbon dioxide released in the bundle sheath cell, you can see over here that the carbon dioxide which is released in the bundle sheath cell now will be fixed by the help of which cycle? By the help of the Calvin cycle or the C3 cycle. And what is the carbon dioxide acceptor of the C3 cycle? It is the RUBP, ribulose 1,5-biphosphate, and this is the final acceptor of carbon dioxide in the C4 plants. Clear? So here you can see that in the mesophyll cells, the initial fixation of carbon dioxide occur where there is formation of oxaloacetic acid, and oxaloacetic acid will be converted into malic acid or aspartic acids. Within the bundle sheath, they will be transported through the help of plasmodesmata, and in the bundle sheath cells, malic acids will be decarboxylated to form pyruvic acid, and aspartic acid will form alanine and oxaloacetic acids, the 4 carbon acids. Okay, and there will be release of carbon dioxide. You can see over here that oxaloacetic acid OAA in reaction with ATP in the presence of enzyme PEP carboxylase will form the pyruvic acid and will release carbon dioxide. So this carbon dioxide which is released in the bundle sheath cells now will be fixed by the help of the Calvin cycle or the C3 cycle. So the fixation of carbon dioxide in the bundle sheath cells takes place by the help of the compound called RUBP or ribulose 1,5-biphosphate, which is secondary or the final acceptor of carbon dioxide in the C4 plants. Clear? So you can see that the fixation of carbon dioxide takes place by the Calvin cycles, and there will be formation of C3 acid, that is phosphoglyceric acids, which will be transported to the mesophyll cells. So fourth one is the regeneration of PEP. So pyruvic acid and PEP formed in the bundle sheath cells. You can see over here that the C3 acid will be formed in the bundle sheath cells and PEP also will be formed. Now those PEP and pyruvic acid which are formed in the bundle sheath cells, they will return to the mesophyll cells. So they will be transported to the mesophyll cells, and alanine will be deaminated through transamination to form the pyruvic acids. Hence pyruvic acid is changed to PEP phosphoenol pyruvic acid, which is the carbon dioxide receptor in C4 pathway. So here you see the reaction that pyruvic acid plus ATP will lead to the formation of PEP phosphoenol pyruvate, which is the carbon dioxide receptor in the C4 pathway. Clear? So here we can see that in C4 pathway that C3 cycle or the Calvin cycle is also occurring in the bundle sheath cells. Clear? So keep in your mind. Now in C3 cycle, 3 ATP were used for the fixation of one carbon dioxide molecule that we have learned. But in C4 pathway, 5 ATP molecules will be utilized. Why? Because in C3 pathway, 3 ATP molecules was being utilized. Okay, but here in C4 pathway, 3 ATP for Calvin cycle and 2 ATP for initial fixation of carbon dioxide takes place. So how many ATP utilized? 5 ATP is utilized. One more time I'm saying that in C3 pathway 3 ATP was utilized for the fixation of one carbon dioxide, but in C4 pathway we require 5 ATP molecules. Why 5? Because in C4 pathway both initial fixation and the final fixation of carbon dioxide takes place. Okay, so therefore for Calvin cycle 3 ATP, and for initial fixation 2 ATP. So 3 plus 2 will be how much? 5 ATP. So 5 ATP will be required in C4 pathway for the fixation of one carbon dioxide molecules. Clear? Okay. Now C4 plants require total 30 ATP molecule to synthesize one molecule of glucose, whereas in case of C3 pathway 18 ATP was required for the synthesis of one molecule of glucose. But here in case of C4 pathway 30 ATP molecules are being utilized for the formation of one molecule of glucose. So the C4 pathway is more energy expensive than compared to the C3 pathway. However, C3 plants lose more than half of the photosynthetic fixed carbon by photorespiration. So therefore C4 pathway is considered to have an adaptive advantage over the C3. If the question comes why C4 pathway is more advantageous over C3 pathway, then you have to say that though the C4 pathway is more energy expensive because it requires 30 ATP molecule compared to 18 ATP in the C3 pathway, but the photosynthetically fixed carbon or the product which is being fixed by the C3 pathway will be used by or will be lost during the process of photorespiration. Okay, since the photosynthetically fixed carbon will be lost by photorespiration during the C3 pathway, so C4 pathway is considered to have a more adaptive advantage over the C3 pathway. But remember that in C3 pathway we require 18 molecule of ATP to fix one molecule of glucose. Okay, but in case of C4 pathway we require 30 molecule of ATP to form one molecule of glucose. Clear? To fix one molecule of carbon in C3 pathway we require 3 ATP molecule, but in C4 pathway to fix one carbon dioxide molecule we require 5 ATP molecules. So this keep in your mind. Alright, so here we have finished the C3 pathway and the C4 pathway. Now let's go through the difference between the C3 and the C4 plants. In table number 13.5, page number is in the page number 13.24. So let's see the difference between the C3 pathway as the C3 plants and the C4 plants. So first one we talk about the anatomy of C3 plant and the C4 plants. Okay, so anatomy of C3 plants. So point number one, we will see that leaves do not possess Cranch anatomy in C3 plants, but Cranch anatomy is present in case of C4 plants. You know what the Cranch anatomy? Okay, where the mesophyll cells will form a ring around the around the bundle sheath cells, around the vascular bundles. Here palisade cell is absent. Now in C3 plants, chloroplasts do not have peripheral reticulum, but in case of C4 plants they have got peripheral reticulum. Okay, chloroplasts are of two types in C3 plants, that is they are monomorphic or one type, but in case of C4 plants there are two types, that they are dimorphic. C3 plants bundle sheath cells usually do not contain chloroplast, but C4 plants bundle sheath cell possess permanent chloroplast. Okay, and C3 plants mesophyll cells perform complete photosynthesis, but in C4 plants mesophyll cells perform only initial fixation. Alright, now let's talk about the Calvin cycle, C3 cycle and the C4 cycle. Now the primary acceptor of carbon dioxide in the C3 cycle is RUBP, whereas in case of C4 cycle the carbon dioxide acceptor is PEP. Okay, phosphoenol pyruvate. Number two, the first stable product in Calvin cycle or the C3 cycle is phosphoglyceric acid, whereas in case of C4 cycle it is oxaloacetic acid. PJ is a 3-carbon compound, whereas OAA, that is oxaloacetic acid, is a 4-carbon compound. Okay. Number three, the C3 cycle operate under very low carbon dioxide concentrations, but C4 cycles operate under very low carbon dioxide. It cannot operate under very low. Like C3 cycle, it cannot operate under very low carbon dioxide concentration, but C4 cycle it can operate under very low carbon dioxide concentration. Okay, in C3 cycle carbon dioxide once fixed is not released back, but in C4 cycle there will be initial fixation and the final fixation of carbon dioxide. So fixed carbon dioxide is released back in the bundle sheath cells, where it is finally fixed and released by the Calvin cycle that we have learned. Okay, now in C3 cycle there is gain of one molecule of glucose by fixing 6 carbon dioxide molecules, but in case of Haslach cycle or the C4 cycle there is no such gain. Okay, fixation of one molecule of carbon dioxide in C3 cycle requires 3 ATP and 2 NADPH molecule, but in case of C4 cycle it requires 5 ATP molecule. Listen this carefully. Okay, it requires 5 ATP molecules. Clear? In C3 cycle it has a slower rate of carbon dioxide fixation, but C4 cycle, it has a faster rate of carbon dioxide fixation. Clear? Now, next one I tell you that point number 10 requires 18 ATP for the synthesis of 1 molecule of glucose in C3 cycle, but in case of C4 cycle 30 molecules of ATP are required for the formation of 1 glucose molecule. Clear? Now, point number 13, let's see that. Calvin cycle occurs in the chloroplast of the mesophyll cells, whereas C4 cycle or the Haslak cycle, it occurs in the chloroplast of bundle sheath cells, as the enzyme for Calvin cycles are absent in the chloroplast of mesophyll cells. Okay, now point number 15, we can say that less efficient in utilizing carbon dioxide, and C4 cycle, they are more efficient in utilizing carbon dioxide. So here we have finished about the mechanism of photosynthesis. So today we have learned about the light independent phase or the dark phase of photosynthesis or the carbon dioxide fixation, and we have learned about the C3 cycle and the C4 cycle also. So this much for today's class. See you in next class, where we'll begin with the new topic on the photosynthesis. Thank you.